Hi everyone. Now, we're going to discuss about Chapter 6, Expression of Biological Information. 6.4, Gene Regulation and Expression, Lac Operon. State the component of operon. Before we discuss further, let's us watch this video to understand what is the importances of gene regulation.
Gene regulation, in the most simplest terms, is the ability of an organism to control or regulate what proteins it makes from its DNA. In our body, we have numerous types of tissues and cells. We have muscle cells known as myocytes, we have cells in our intestines known as enterocytes, and the cells in our skin known as keratinocytes. All these cells are different in shapes, sizes, and functions as well. The myocytes' main job is muscle contraction, enterocytes produce enzymes for food digestion, and keratinocytes form a barrier known as skin. All these cells contain the same DNA in them. However, they are different in sizes, structure, and function due to the different proteins they produce. As most of you may know, our DNA is made up of thousands of nucleotide bases which are paired, and these are the basic units of DNA. The human DNA contains over 3.1 billion such base pairs. These base pairs are arranged in groups known as genes. Now, if you take out the DNA from the nucleus of a muscle cell, a skin cell, and a cell from the intestine, they would contain the same 3.1 billion base pairs of nucleotides arranged in the same sequence. But despite having the same DNA in them, they are completely different, and this is because of the different proteins they produce. This is all possible due to the ability of the cell to control or regulate what proteins it makes from its DNA.
What is operon? Actually, operon is a genetic regulatory system found in bacteria, in which genes coding for functionally related proteins are clustered along the DNA. There are three components of operon, which is promoter, short segment of DNA, to which the RNA polymerase binds to initiate transcription. An operator, short segment of DNA, found between the promoter and the structural genes. And structural genes, single unit of messenger RNA, mRNA, to be transcribed.
Of gene expression. Now, to keep it very simple, the central dogma basically says that the information is copied from DNA to RNA, which is known as transcription, and from RNA to proteins, which is known as translation. So the gene regulation can work at multiple levels in this model of gene expression, but the most common point where the gene regulation operates is at the level of transcription, the point where the information on the DNA is copied onto RNA. Now, like we discussed earlier in this video, we have structural genes that code for proteins, and then we have regulatory DNA which regulates the proteins the structural DNA makes. Let's look at how this regulatory portion of DNA is usually arranged and what is the most common mechanism in gene regulation. Now, suppose we have two genes on this chromosome that code for protein A and protein B. Just before these two protein coding genes, we have the regulatory DNA that is shown in blue, which controls these genes. The regulatory portion of DNA is usually arranged into a regulatory sequence that is shown in light blue and regulatory gene, which is shown in dark blue. The regulatory sequence is usually a few hundred to thousand base pair long sequence that lies just behind the main protein coding genes, and it is the site of attachment of the enzyme RNA polymerase. This enzyme copies the information on the genes A and B onto an mRNA, that is the process of transcription. The main regulatory gene usually lies a few thousand base pairs behind the protein coding genes, and this regulatory gene synthesizes a protein known as the regulatory protein. The regulatory protein goes on and controls the binding of enzyme RNA polymerase to the regulatory sequence, increasing it or decreasing it depending upon the situation. So this is the basic model of how gene regulation works at the level of transcription. We have regulatory genes that control the rate of transcription of the protein coding genes, also known as the structural genes. The regulatory sequence is in turn divided into an operator, promoter, and many other parts, the details of which we will talk about later in this video. But you can just remember that depending upon whether the cell needs these proteins or not, the regulatory gene synthesizes regulatory protein that can block or promote the binding of RNA polymerase to the regulatory sequence, and thus decreasing or increasing the synthesis of proteins from the structural genes. This is obviously different for different types of genes, but I hope I am able to make.
To discuss about the Lac Operon, we have to study the experiment by Jacob and Minot. They have demonstrated the function of operon, using E. coli, to regulate the metabolism of lactose. Now, let us discuss about Lac Operon. Lac Operon is a cluster of genes, consisting of operator, promoter and structural genes, which is LAC-C, LAC-Y and LAC-A, that codes for enzymes used to metabolize lactose in E. coli.
In a better way. So this is Lac Operon. This is basically a small piece of bacterial DNA and consists of a few important genes that code for the enzymes that allows the bacteria to break down lactose. The Lac Operon can be broadly divided into two important regions: the structural genes and the regulatory portion. The structural genes code for the enzymes, whereas the regulatory region of Lac Operon controls or regulates these structural genes. The regulatory region of Lac Operon allows the bacteria to essentially switch on or off the structural genes depending upon whether lactose is available or not. There are three important structural genes in the Lac Operon that are known as LAC-Z, LAC-Y, and LAC-A. LAC stands for lactose, the sugar for which these genes make the enzymes for, and Z, Y, A are arbitrary alphabets used to denote the names of these genes. The gene LAC-Z codes for the enzyme beta-galactosidase. As you may know by now that the sugar lactose is a disaccharide, meaning it is made up of two simple sugars, glucose and galactose, joined by a glycosidic bond. The enzyme beta-galactosidase breaks the bond between glucose and galactose, thus converting lactose, which is a disaccharide, into two simple sugars. The gene LAC-Y codes for the protein beta-galactoside permease, which is basically a tube-like protein that fits into the cell membrane of the bacteria and allows more and more lactose to be pumped into the cell. That is why it is known as beta-galactoside permease, because it increases the permeability of the cell membrane to beta-galactosides, which includes sugars like lactose. The third structural gene in the Lac Operon codes for beta-galactoside transacetylase, an enzyme that transfers an acetyl group from acetyl-CoA to beta-galactosides. Its function is not fully understood in the metabolism. So these are the three structural genes of Lac Operon. The region in blue is the regulatory portion of Lac Operon, and it is divided into many sub-regions. We have the LAC-I gene, which codes for the protein repressor. We also have a promoter, an operator. We also have a cap site, and at the end of Lac Operon, we have a terminator. At first, these may seem difficult, but I'll try to explain them in such a manner that you'll remember the regulation of Lac Operon in a better way. So, we have our enzyme RNA polymerase that has to copy the information of three structural genes of Lac Operon onto an mRNA. Now, RNA polymerases always bind at a site located just behind the structural genes, and this is known as the promoter. Just in front of the promoter, we have our operator, and you can remember that the operator as a switch which can be turned on or off. When it is turned off, the RNA polymerase cannot move forward on the DNA, and hence it cannot read what's written on it, so the genes are not transcribed. When it is turned on, the RNA polymerase on the promoter can now move forward, and the genes ZYA are transcribed into a mRNA, which is then used to make proteins. Now, I remember that while learning this, I always used to forget whether the RNA polymerase binds with the promoter or the operator. Well, you can memorize this by just remembering that promoter starts with P, so it binds with RNA polymerase, which also starts with P. P for promoter and P for polymerase. Now the next thing to understand here is what turns the operator on or off, and this is basically the whole concept of gene regulation in Lac Operon. Now here comes the role of a gene that is located a few thousand base pairs behind the promoter, and this is known as the Lac I gene, where I is standing for inducible. So this gene continuously synthesizes a protein that is known as a repressor. The repressor is a claw-like protein which can exist in two different forms. When the repressor attaches its claws to the operator, this is known as the bound state, and this is when the red light is turned on by the operator, meaning it is turned off. The RNA polymerase cannot move forward on the DNA because there is literally a roadblock sitting in its way that prevents it from moving forward on the DNA, and the genes are then not transcribed. And you must also note here that this is usually the normal state of Lac Operon 90% of the times.
In presence of glucose, lactose will not digest, or absent, so the Lac Operon switches off. This is due to the binding of repressor protein to the operator and block promoter site. RNA polymerase cannot bind to the promoter to start transcription of the structural gene. In presence of lactose, it will convert it to allolactose, catalyzed by transacetylase. Allolactose then bind to repressor protein and change the conformation of repressor protein. Thus, repressor protein cannot bind to operator and Lac Operon switch on. RNA polymerase will binds to the promoter and transcribes the structural gene into mRNA. mRNA is translated into three structural enzymes, which is beta-galactosidase, that catalyze the hydrolysis of lactose into glucose and galactose. Permease catalyze the transport of lactose into E. coli. And transacetylase catalyze the transfers of an acetyl group from acetyl-CoA to beta-galactosidase.